future of technology is a vast and exciting frontier. We stand at the precipice of incredible advancements, breakthroughs that promise to reshape our world. Among these, human cloning holds immense potential and debate. Imagine a world where organ failure is a distant memory, where genetic diseases can be eradicated before birth. This is the promise of cloning technology, but it comes with ethical challenges and potential pitfalls. The ability to create a genetically identical copy of a human being raises profound questions. Is it ethically permissible to manipulate the building blocks of life in this way? What are the potential consequences of such power? These are real and urgent concerns. We must grapple with them as we venture further into this uncharted territory. Join me as we explore the science, benefits, and ethical implications of human cloning. Human cloning, once science fiction, is now a tangible possibility. With this power comes profound responsibility. What does it mean to be human? Is our identity solely in our genes? These are real-world dilemmas with far-reaching consequences. We must engage in thoughtful dialogue about this powerful technology. Proceed with caution, guided by wisdom and compassion. The rapid pace of technological advancement often outstrips our ability to fully comprehend its ethical implications. Human cloning is a prime example. The ethical dilemmas it poses are intricate. We are venturing into uncharted territory. One pressing concern is exploitation. Will cloning be accessible to all or just the wealthy? Could individuals be cloned without consent? These are disturbing possibilities. Another issue is the impact on human relationships. What happens to the traditional family structure? How will we define parenthood or siblinghood? These questions are fundamental to our understanding of human connection. The ethical frontier of human cloning is vast and complex. There are no easy answers. We must engage in open and honest dialogue, develop robust legal and regulatory frameworks. The future of human cloning is in our hands. Ensure this technology benefits all of humanity. Embrace this task with humility and determination. Human cloning, once a term of fear and fascination, is moving from science fiction to reality. It represents a leap in manipulating life's building blocks, raising profound questions. At its core, human cloning creates a genetically identical copy of a human. It challenges our understanding of identity and individuality. Cloning holds potential for medical breakthroughs, but raises ethical concerns. The two roads of cloning. Reproductive cloning captures the imagination with its potential to create a genetically identical copy of a human being. Imagine a world where parents struggling with infertility could have children who are genetically related to them, or where individuals could, in theory, create copies of themselves. The implications are both exhilarating and unsettling, raising questions about identity, individuality, and the very definition of family. Therapeutic cloning, while less sensationalized, holds immense promise for medical advancements. By creating cloned embryos, scientists can harvest embryonic stem cells, which have the unique ability to develop into any type of cell in the body. This has the potential to revolutionize medicine, offering new treatments for a wide range of diseases and conditions, from Parkinson's disease and Alzheimer's disease to spinal cord injuries and heart disease. The ethical considerations surrounding reproductive and therapeutic cloning are distinct and complex. Reproductive cloning raises concerns about the psychological well-being of cloned individuals, the potential for coercion and exploitation, and the societal implications of fundamentally altering human reproduction. Therapeutic cloning, while ethically less fraught, still sparks debate about the moral status of embryos and the potential for slippery slopes leading to reproductive cloning. The scientific community and society as a whole are grappling with these complex issues, recognizing the need for careful consideration and robust ethical guidelines. The potential benefits of cloning technologies are undeniable, but so are the potential risks. Navigating this uncharted territory requires a delicate balance between scientific progress and ethical responsibility, ensuring that we harness the power of cloning for the betterment of humanity while safeguarding against its potential pitfalls. A sheep named Dolly, a giant leap for cloning. The world watched in awe and apprehension in 1996 when news broke of Dolly the sheep, the first mammal successfully cloned from an adult cell. This groundbreaking achievement, the culmination of years of research by scientists Ian Wilmot and Keith Campbell at the Roslin Institute in Scotland, marked a paradigm shift in our understanding of biology and ignited a global debate about the ethics and implications of cloning. Dolly's birth was not just a scientific marvel, it was a cultural phenomenon. It captured the public imagination, sparking both excitement and fear about the possibility of cloning humans. Suddenly, what was once relegated to the realm of science fiction seemed tantalizingly within reach, forcing society to confront the potential consequences of this newfound power. Dolly's legacy extends far beyond her relatively short lifespan. Her birth ushered in a new era of cloning research, inspiring scientists around the world to explore the potential of this technology in various species. It also led to a surge in public interest and investment in stem cell research, as scientists recognized the potential of cloning for therapeutic applications. However, Dolly's story also served as a cautionary tale. She suffered from health problems, including premature arthritis and lung disease, raising concerns about the long-term health and well-being of cloned animals. This highlighted the need for further research and ethical considerations before cloning could be considered a safe and viable option for humans. Dolly's life and legacy continue to shape the debate surrounding cloning, reminding us of both the immense potential and the potential pitfalls of this powerful technology. The dance of DNA deciphering somatic cell nuclear transfer. The creation of Dolly the sheep was made possible by a groundbreaking technique known as somatic cell nuclear transfer, or SCNT, a process that lies at the heart of cloning. This intricate cellular dance involves replacing the nucleus of an egg cell with the nucleus of a somatic cell, which is any cell in the body other than sperm and egg cells. This effectively reprograms the egg cell, transforming it from a blank slate into a cell with the genetic blueprint of the donor organism. The process begins with the extraction of a somatic cell from the donor organism. This cell contains all of the organism's genetic information within its nucleus. Meanwhile, an egg cell is obtained from a female of the same species, and its nucleus, which contains only half of the genetic material, is carefully removed. The nucleus of the donor somatic cell is then injected into the enucleated egg cell. The egg cell, now carrying the donor's genetic material, is stimulated to divide and develop into an embryo. This embryo is then implanted into the uterus of a surrogate mother, where it continues to develop until birth. The offspring resulting from this process is a genetic clone of the donor organism. This is because the offspring's DNA comes not from the combination of sperm and egg DNA, as in natural reproduction, but solely from the nucleus of the donor somatic cell. While SCNT has been successfully used to clone various animals, including sheep, cows, pigs, and even monkeys, it remains a complex and inefficient procedure. Beyond the petri dish, the potential and perils of cloning. The science of cloning, particularly the intricacies of somatic cell nuclear transfer, has captivated the scientific community and ignited the imaginations of people worldwide. The potential applications of this technology extend far beyond the realm of simply replicating existing life forms. It holds the promise of revolutionizing medicine, agriculture, and even conservation efforts, but it also presents us with profound ethical dilemmas that we must navigate with caution and foresight. In the field of medicine, cloning offers tantalizing possibilities for organ transplantation, regenerative medicine, and the treatment of genetic diseases. Imagine a future where patients in need of organ transplants no longer have to rely on the scarcity of organ donors, but could instead receive perfectly matched organs grown from their own cells. This could potentially save countless lives and alleviate the suffering of millions worldwide. Beyond organ transplantation, cloning holds promise for treating debilitating diseases like Parkinson's, Alzheimer's, and spinal cord injuries. The ability to generate specific cell types from a patient's own DNA could revolutionize the field of regenerative medicine, offering new hope for those suffering from currently incurable conditions. However, the potential benefits of cloning must be carefully weighed against its ethical implications. The creation of human embryos solely for research purposes raises moral concerns, while the prospect of reproductive cloning raises even more profound questions about human dignity, identity, and the very meaning of parenthood. As we venture further into this uncharted territory, it is imperative that we proceed with caution, guided by ethical principles and a deep respect for the sanctity of life. Advancements and achievements. Cloning technology is evolving, marked by progress and occasional leaps forward. Scientists worldwide are refining techniques and exploring new avenues. Recent advancements bring us closer to realizing cloning's full potential. Significant improvements in somatic cell nuclear transfer, or SCNT, have been made. SCNT involves transferring a donor cell nucleus into an enucleated egg cell. Fine-tuning parameters has increased SCNT success rates in various species. In agriculture, cloning replicates prized livestock, improving herd quality. A global tapestry of cloning research. Cloning technology is a global endeavor, spanning continents and disciplines. From North America and Europe to Asia, scientists work tirelessly to unlock cloning secrets. This global collaboration fosters a rich tapestry of research, advancing our understanding of this promising field. From animals to humans, bridging the gap. While the ethical considerations surrounding human cloning remain a topic of intense debate, the advancements made in animal cloning have undoubtedly paved the way for potential applications in humans. Each successful cloning of an animal species, from Dolly the sheep to the more recent cloning of primates, brings us closer to understanding the complexities of this technology and its potential implications for human health and well-being. The successful cloning of primates, our closest biological relatives, marked a significant milestone in cloning technology. In 2018, researchers at the Chinese Academy of Sciences made headlines worldwide when they announced the birth of Zhongzhong and Huahua, two genetically identical long-tailed macaques using the SCNT technique. This breakthrough demonstrated that primates, with their complex biology and developmental processes, could be successfully cloned, opening up new avenues for research into human diseases and potential therapies. The knowledge gained from animal cloning studies has been instrumental in refining cloning techniques and identifying key factors that influence success rates. For instance, researchers have discovered that the type of donor cells used, the methods used to manipulate the egg and donor cells, and the culture conditions used to support embryo development can all significantly impact the outcome of the cloning process. These findings have important implications for potential future applications in humans, as they highlight the need for careful optimization of the cloning procedure to ensure both safety and efficacy. Moreover, animal cloning has provided valuable insights into the long-term health and well-being of cloned individuals. While early concerns about premature aging and health problems in cloned animals have proven to be largely unfounded, ongoing research continues to monitor the health of cloned animals throughout their lifespans. These studies are crucial for assessing the potential risks and benefits of cloning technology, and for developing ethical guidelines for its use in both animals and, potentially, humans in the future. Tackling the challenges of cloning. Despite remarkable progress, cloning efficiency remains a significant challenge. Success rates are low, often requiring hundreds of attempts for one viable clone. The reprogramming process is complex and delicate. Researchers are exploring new techniques to improve efficiency. Continued improvements will pave the way for wider applications. The ethical imperative balancing progress with responsibility. As cloning technology advances, it is imperative that we proceed with caution, guided by a strong ethical framework. The potential benefits of cloning are undeniable, but so too are the potential risks. One of the most pressing ethical concerns is the potential for harm to cloned individuals. Advancements have reduced risks, but further research is needed for long-term health. The psychological impact of being a clone requires careful consideration. Beyond individual well-being, there are broader societal implications. Cloning could exacerbate social inequalities if access is limited to the wealthy. The commodification of human life and potential exploitation are complex ethical dilemmas. Addressing these concerns requires open dialogue and robust regulatory frameworks. Public education and engagement are crucial for informed decision-making. Let us proceed with caution and courage, guided by responsibility and respect for life. A new dawn for medicine cloning's therapeutic promise. The prospect of human cloning has sparked both awe and apprehension, igniting passionate debates about its ethical implications. Human cloning, specifically therapeutic cloning, offers a tantalizing glimpse into a future where some of humanity's most formidable medical challenges could be overcome. Imagine a world where organ transplantation is no longer hampered by the chronic shortage of donors. This is one of the most promising applications of human cloning, the ability to create perfectly matched organs for transplantation. Organ regeneration, a future without scarcity. The current system of organ transplantation is fraught with limitations. The demand for organs far outstrips the supply. Human cloning offers a transformative solution. Imagine a future with perfectly matched organs grown from your own cells. This would alleviate suffering and free up healthcare resources. Continued research and investment are essential. Stem cell therapy, a revolution in regenerative medicine. Stem cells hold immense promise for repairing damaged tissues and treating debilitating diseases. Their unique ability to differentiate into specialized cell types is at the heart of this revolution. The possibilities are transformative, offering hope to millions worldwide. Editing our genetic destinies. Inherited diseases have plagued humanity since the dawn of our species. These conditions can lead to a lifetime of suffering, disability, and premature death. Gene editing like CRISPR-Cas9 offers hope for eradicating these diseases. These tools allow scientists to correct genetic defects at their source. Combined with human cloning, gene editing can prevent inherited diseases. The process involves correcting the genetic defect before implantation. The resulting child would be born free of the inherited disease. The ethical considerations are complex, but the benefits are undeniable. Imagine a world where inherited diseases are history. This is the promise of human cloning combined with gene editing. A lifeline for endangered species, cloning for conservation. Human activities drive habitat loss and species decline, making conservation crucial. Cloning offers a lifeline to endangered species on the brink of extinction. Imagine reversing extinction, bringing back lost species. Cloning technology has already cloned endangered animals, offering hope. In 2003, a banteng was cloned from frozen skin cells at the San Diego Zoo. This showed cloning's potential for preserving genetic diversity. Cloning uses somatic cell nuclear transfer, like with Dolly the sheep. A skin cell's nucleus is transferred into an enucleated egg cell. The embryo is implanted into a surrogate mother. Cloning can't solve the biodiversity crisis alone, but is a valuable tool. Preserving genetic legacy keeps the possibility of reintroduction into the wild. The moral labyrinth. Navigating the ethics of human cloning. Human cloning presents us with a moral labyrinth, a complex web of ethical considerations. The ability to create life forces us to confront fundamental questions about human life and scientific boundaries. We must proceed with caution and courage, guided by ethical principles. Identity's echo. The question of individuality in a cloned world. The prospect of human cloning challenges our understanding of identity and individuality. What does it mean to be unique in a world of genetic duplicates? These questions have profound implications for individuals in a future shaped by cloning. Cloned individuals might face challenges in developing a strong sense of self. Being a genetic replica could lead to feelings of inadequacy or a struggle to establish a unique identity. Ensuring the psychological well-being of cloned individuals is crucial. Individuality is shaped by genes, experiences, and personal choices. Clones, while sharing the same genetic blueprint, would have unique experiences and relationships. Recognizing this individuality is crucial for ethical treatment. The question of individuality extends to familial and societal relationships. How would family dynamics change with genetic replicas? Would cloned children be seen as individuals or extensions of their parents' desires? Society must grapple with these ethical implications of human cloning. Playing God? The societal dilemma of artificial human creation. Human cloning forces society to confront the implications of assuming a role once reserved for the divine. The ethical debate questions if we have the right to play God. For many, creating human life artificially represents a dangerous overstepping of boundaries. They argue human life is sacred and should not be subject to scientific experimentation. Others see cloning as a tool to alleviate suffering and enhance human potential. This debate is complicated by differing cultural perspectives on playing God. The cloned commodity, ethical concerns of organ harvesting. Cloning embryos for organ harvesting raises profound ethical questions. The technological possibility forces us to confront these implications. Creating life solely for instrumental purposes is morally complex. Proponents argue it could save lives by providing matched organs. Opponents contend it reduces human life to a means to an end. The ethical dilemma highlights the need for careful consideration. The slippery slope safeguarding against the misuse of cloning. The potential for misuse of human cloning is a significant concern. Robust ethical guidelines and stringent regulations are essential. Cloning for eugenic purposes could have dire consequences. We must ensure cloning is never used for discrimination or oppression. Protecting the vulnerable from exploitation is crucial. The long-term health effects of cloning are still unknown. The legal landscape of human cloning is diverse globally. No universal stance exists, resulting in varied laws. Europe enacts strict bans, especially Germany due to its history. Some countries allow limited research under strict oversight. Ambiguous laws in some regions raise ethical concerns. The shadow of bans navigating international agreements. The lack of a unified global stance on human cloning has led to calls for international agreements or treaties to establish common ethical and legal frameworks. One of the most prominent international instruments addressing human cloning is the United Nations Declaration on Human Cloning, adopted by the General Assembly in 2005. This non-binding declaration calls upon member states to prohibit all forms of human cloning that are incompatible with human dignity and the protection of human life. The Council of Europe adopted the Convention on Human Rights and Biomedicine, also known as the Oviedo Convention, which prohibits the creation of human embryos for research purposes. Despite these efforts, the international community remains divided on the issue of human cloning. Voices of reason, bioethics, and public opinion in the cloning debate. The ethical complexities of human cloning have sparked intense debate not only within scientific and legal circles, but also among the general public. Bioethics committees, composed of experts from various disciplines, provide guidance to policymakers and researchers, ensuring that ethical considerations are given due weight in the development and application of cloning technology. Public opinion on human cloning is far from monolithic, reflecting a spectrum of views shaped by individual values, religious beliefs, and levels of trust in science and technology. The role of public engagement and education in shaping the ethical and legal landscape surrounding human cloning cannot be overstated. Open and transparent dialogue, informed by scientific evidence, ethical considerations, and diverse cultural perspectives, is crucial to fostering a well-informed public. Peering into the future, envisioning cloning's trajectory. Gazing into the crystal ball of technological advancement is always a tantalizing, if uncertain, endeavor. One possibility is that human cloning will follow a similar trajectory to other groundbreaking technologies, initially met with fear and resistance, but gradually gaining acceptance as its benefits become clearer. Just as in vitro fertilization was once a controversial procedure, but is now a widely accepted fertility treatment, human cloning, particularly for therapeutic purposes, may eventually become a routine part of medical practice. The public perception of human cloning, influenced by religious perspectives, philosophical debates, and portrayals in popular culture, will play a significant role in shaping its future. The ability to create genetically identical human beings could have profound implications for our understanding of individuality, the dynamics of family relationships, and even the very fabric of society. Medical marvels, a future transformed by cloning. The potential medical benefits of human cloning are perhaps the most compelling argument in its favor. One of the most promising applications is the regeneration of organs and tissues using a patient's own cells, eliminating the risk of rejection and the need for lifelong immunosuppressant drugs. Human cloning could also pave the way for personalized stem cell therapies, tailored to an individual's unique genetic makeup. Cloning technology could enable the creation of patient-specific stem cell lines, increasing the likelihood of successful treatment and minimizing the risk of immune rejection. Furthermore, human cloning, combined with gene editing technologies like CRISPR-Cas9, could potentially eradicate inherited diseases, preventing them from being passed down to future generations. The ethical tightrope. Navigating the dilemmas of a cloned future. The potential benefits of human cloning are undeniable, but they come intertwined with a complex web of ethical dilemmas that demand careful consideration. One of the most pressing ethical concerns is the potential for harm to cloned individuals, both physically and psychologically. The right to an open future, free from predetermined expectations or comparisons to the original, is a fundamental human right that must be carefully guarded. The creation of human life solely for instrumental purposes, such as organ harvesting, raises profound ethical concerns. Ensuring equitable access to any potential benefits of cloning technology is crucial to prevent further widening the gap between the haves and the have-nots. The social fabric reshaping human relationships and identity. The advent of human cloning has the potential to reshape the very fabric of society, challenging our understanding of family identity and the relationships that bind us together. The traditional family structure based on biological ties and the union of sperm and egg could be fundamentally altered in a world where genetic replicas could be created. Human cloning could also challenge our understanding of identity, both individual and collective. The potential for cloning to be used for eugenic purposes, attempting to create a superior race, or eliminate undesirable traits from the human gene pool, raises chilling ethical concerns. Safeguarding against such misuse is paramount, requiring vigilance, ethical awareness, and robust legal frameworks that prioritize the value and equality of all human beings. Governing the future. The evolving landscape of cloning regulations. The rapid pace of advancements in cloning technology presents significant challenges for policymakers and regulators worldwide. As this technology continues to evolve, the regulatory landscape must adapt to keep pace, striking a delicate balance between fostering innovation and safeguarding against potential harms. One of the key challenges is establishing clear and consistent regulations across borders. International cooperation and harmonization of regulations are essential to prevent the exploitation of vulnerable populations and ensure that human cloning, if pursued, is conducted ethically and responsibly. Public engagement and education are crucial for fostering informed decision-making and ensuring that regulations reflect the values and aspirations of society as a whole. The unfolding tapestry of life. We've journeyed together to the frontiers of science, peering into the intricate mechanisms of life itself and grappling with the profound implications of human cloning. From the laboratories where cells dance to the music of DNA to the halls of government where ethical debates rage, we've explored the multifaceted dimensions of this revolutionary technology. Human cloning, in its essence, represents our species' bold attempt to grasp the very threads of this tapestry, to understand its intricate patterns, and perhaps even to contribute our own designs. The ethical considerations surrounding human cloning are as intricate as the DNA double helix itself. The decisions we make today about human cloning will have far-reaching consequences for generations to come, shaping the future of our species and our relationship with the natural world. A call to conscious innovation. The future of human cloning is not preordained. It is a future we shape through our choices and actions. The potential benefits of human cloning are undeniable, but realizing these benefits requires conscious innovation. This journey demands collective wisdom. Open dialogue is essential for aligning cloning technology with humanity's values. Our shared genetic legacy. As we conclude our exploration of human cloning, it is important to remember that this technology, for all its complexities and controversies, is ultimately about our shared genetic legacy. The ability to manipulate DNA to create life in our own image is a testament to the remarkable ingenuity of our species and our insatiable thirst for knowledge. But it also serves as a profound reminder of our interconnectedness, of the shared genetic heritage that binds us together as a species and connects us to the vast web of life on Earth. The decisions we make about human cloning will have implications not only for ourselves, but for future generations who will inherit the consequences of our choices. Will we use this technology wisely to alleviate suffering, enhance human potential, and preserve the diversity of life on Earth? Or will we succumb to fear, prejudice, or the allure of unchecked ambition, risking the creation of a future where human dignity is compromised and the very essence of what it means to be human is called into question? The answers to these questions lie not in the realm of science alone, but in the hearts and minds of individuals who will ultimately shape the course of this technology. Education, ethical reflection, and open dialogue are essential tools for navigating this complex terrain. By engaging in thoughtful deliberation, listening to diverse perspectives, and prioritizing the well-being of all, we can strive to create a future where human cloning, if pursued, is used responsibly and ethically, enriching our lives and preserving the precious gift of life for generations to come.